everybody. My name is Molly Hunt, and I'm one of the student assistants at the Orton Geological Museum. I'm so excited that you are spending your Friday afternoon with us, as this week is National Earth Sciences Week. On Wednesday was National Fossil Day, and so we have brought you a special presentation from Dr. Loren Babcock, a professor in the School of Earth Sciences, and also the director of the Orton Geological Museum. He will give a presentation on some really neat work with trilobites. And so I will take it over to him, and I hope you enjoy his presentation. If you have questions throughout it, you can feel free to put those in the comments below on Facebook, or if you're on our Zoom, you can also put those in the chat box. And at the end, we'll let him answer those. So Dr. Babcock, I'll let you go.
All right, thanks, Molly, and thanks everyone for joining us. It's nice to have the opportunity to talk to all of you. For those who don't know me, I have a variety of research interests. They commonly revolve around fossilization processes, around the early development of animal life on this planet, and especially a lot of my work involves major evolutionary events in Earth history, and they include things like the so-called Cambrian explosion, or other sorts of times in Earth history when life went through sort of critical stages. And what I'd like to talk to you about today is something that merges on a couple of these topics. And we'll talk a little bit about some Cambrian fossils, and the processes of fossilization of some of those, and some of what we refer to as exceptional fossil preservation, where we get the non-biomineralized parts of organisms preserved. Under ordinary circumstances, fossils normally comprise so-called hard parts, things like bones, teeth, and shells. In exceptional fossil preservation, we get other parts of creatures' anatomy being preserved, things like skin or feathers or hair or the internal guts, things like this. And that's what I want to talk about today is examples of some exceptional fossil preservation coming from some of the earliest known animal fossils, and the way that these exceptionally preserved remains were discovered was through a combination of modern technologies, including scanning electron microscopy and also X-ray CT scanning. And so let me share my screen with you and show you some of what we've been doing here. Okay, I hope you can all see that now. And so this is actually a repeat of a talk that I gave in Riverside, California over the past summer at the North American Paleontological Convention. This is a talk that was put together jointly with some of my students or former students, plus one of our technicians. And so Jesse Castigar was a former student of mine, Jenny Patea, a former student, and Kira Eaton as well. And I think at least one of them is on the line now. And then Julie Sheets particularly was helpful with scanning electron microscopy work. And this, what we're talking about here is going to be similar in some ways to what you may have heard a couple of months ago when Prescott Veda spoke about CT scanning. He was discussing some CT scans of Devonian fossils from Ohio and adjacent areas. What I'm going to talk about is what we've discovered through similar means on the CT scanner for much earlier material, mostly coming from Western and Southeastern North America. So what we'll talk about today then is examples of some 3D preservation of internal soft parts or of other non-biomineralized tissues in some Cambrian age fossils from North America. And these ones are special in that they come from concretions or sort of rounded carbonate masses or silica masses around what presumably was decaying organic material at the time of sediment deposition. And the term Lagerstätten, for those who are not used to that term, the Lagerstätten are deposits where we find remains of exceptionally preserved fossils. Okay, so with that, let's take a look at some of the information related to the topic. So, just as a bit of background, when we look at Cambrian deposits, essentially on a global scale, we find that concretions are extremely common in certain intervals. An example here from a photograph here is a professor from the University of Cincinnati. This is Carl Brett here, examining a very large concretion in the alum shale in Sweden. And this is an example of a carbonate concretion, very similar to the large carbonate concretions that we see in the Devonian of central Ohio or northeastern Ohio, for example. And these particular concretions, these giant concretions in Scandinavia, are often referred to as so-called Orsten. We find similar ones in South China and in some other places. Sometimes they yield exceptionally preserved fossils. Another style of concretion formation that we see in the Cambrian that's present in a few locations is where we see silica in the form of quartz, actually microcrystalline quartz, which we call chalcedony, forming concretions around organic material. We see these especially along the Georgia-Alabama border area, and also in Spain. Well, some of the best known concretions, even though you may not have thought of them in quite this way, but probably the best known concretions that there are coming from Cambrian deposits are calcitic in nature. They're formed of calcium carbonate, and they have a very special sort of a pattern to them, which is called a cone and cone fabric. And I'll show you this a little bit later on. And so what I'm pointing to here is three examples of trilobites. These are the very common Elrathia that come from the Wheeler Formation of Western Utah. And each one of these specimens is preserved on a tiny little concretion. The margins of the concretion represent, the margins of the concretion coincide with the edges of the trilobite exoskeleton. And from other work that we've published, we know that the margins of those concretions actually correspond to the margins of the so-called biofilm, or the slime, the decay halo that develops on the organisms shortly after they die and begin the decay process. And so these concretions, in the shapes of those concretions, the sizes of these concretions, is related to what we refer to as the decay halo, or the biofilm that was responsible in part for decaying organic material. So what I'd like to talk about in the few minutes that I have is exceptional preservation in some of these calcite concretions that come from the Wheeler Formation, or the next unit up, which we call the Margin Formation in Utah, and also from some of these siliceous or quartz concretions that come from the Conasauga Formation in Alabama and Georgia. And what is really special about the preservation in these concretions is not simply that we have exceptionally preserved parts, but that the preservation within these concretions differs from the preservation that we typically find in shales, where fossil remains tend to be compressed. Inside the concretions, we commonly find fully inflated three-dimensional preservation. And we get, as you will see, we get not just macroscopic fossils like trilobites, whole trilobites being preserved, but we also get microscopic fossils being preserved, including things like bacteria and fungal strands. And so these concretions yield information about the fossilization process, about the decay process and about paleoecology that are basically unobtainable by almost any other means. Now, it's a fascinating sort of thing. When you look at Cambrian deposits on a worldwide scale, and I suspect this is probably true with almost any interval of geologic time, but the time I've spent most of my work looking at is in the Cambrian. When we look at Cambrian deposits on a global scale, we find that there is a fairly consistent pattern of positioning of concretionary units in the stratigraphic record, and the positions of those concretions is linked to positions in the global sea level history. And so coincidentally, the positions of these deposits of exceptional fossil preservation, or Lagerstätten, also seem to be linked to positions in the global sea level record. And sometimes the positions of the concretions are the same as the positions of some of the Lagerstätten. And as we've shown, I think rather convincingly, we are fairly confident now, I think, in saying that much of what was driving global sea level changes through the early part of the Paleozoic, including in most of the Cambrian, was changes in glaciation, in deglaciation, in the southernmost continental area, which is what we call Gondwana. And so we can actually link global climatic changes and sea level changes, not just in time intervals like the present or in the Pleistocene, or in other time intervals like the Devonian, for example, but we can also identify similar sea level patterns related to glaciation and deglaciation and global climatic change, going all the way back to at least the Cambrian. And so what I have in this diagram here is first of all, off to the left is chronostratigraphic information. The time intervals we're talking about here are within the upper half, or just a little less than the half, a little less than half actually of the Cambrian. We're talking about the Maulingian and Thuringian epochs, or in chronostratigraphic terms, we call them series of the Cambrian. And we're talking about finding concretions in primarily the Woollyuan, the Drumian, Guzhangian, and then the Jiangshanian ages, or in chronostratigraphic terms, stages of the Cambrian. This column here provides a global compilation of the eustatic record beginning in the Woollyuan, and sea level increases to the right here, and then sea level decrease to the left. And so what we're looking at is the positions of these concretionary intervals indicated by these big black dots, which are supposed to be concretions, in some of these positions. We find a fair number of concretions, for example, in the Spence Shale, the Burgess Shale, the Kaili of China. For example, I think most people are familiar with those units. We see concretions in the Wheeler Formation. These are probably the most famous concretions on the planet. And the Wheeler concretions coincide in time almost exactly with those that are forming in the Conasauga Formation of Georgia and Alabama. We see concretions in the overlying unit in Utah. The unit overlies the Wheeler, that's the Margin Formation. And we see them again up in the Weeks to a certain extent. And then, of course, we have the very famous and large concretions in the Alum Shale of Scandinavia, and similar ones in the Huaqiao Formation of South China. All of these positions here are also deposits of exceptional preservation. And so we often find exceptionally preserved fossils in the shales or the limestones, or whatever units, whatever lithologies they happen to be present in these intervals, at least in these stratigraphic deposits, to a lesser extent here, I suppose. But we do find exceptionally preserved fossils also in the concretions. And when they're in the concretions, they are commonly preserved in 3 dimensions. So let's take a look at the setup in Western Utah, where a lot of this material comes from. And this is a sort of a summary stratigraphic diagram for the Merolingian of Western Utah. Here's a map showing where Utah is if you can't remember. And what we have here are various indicators of biostratigraphic zonation. And over to the right, we have the lithostratigraphic units. The units, or the intervals within the units where concretions are common, tend to be sort of in this portion of the Wheeler Formation and in this lower portion of the Margin Formation, which overlies the Wheeler. And this is a fairly typical example of a trilobite. This one happens to come out of the Margin Formation. And what you see is very clearly here is the margins of the concretion. And again, as I said, this, as we now know, corresponds to the margin of the decay halo or the biofilm that surrounded the trilobite corpse at the time that it was decaying. And then we see a beautifully preserved, outstretched, complete trilobite preserved on the surface of this concretion. This is very typical of the kinds of concretions we find in the Wheeler and the Margin. We find the exceptionally preserved fossils usually on the outer surfaces. And just to sort of hammer this point home a little bit further here, in the Wheeler and Margin formations, we find a large number of species that are preserved in exceptional condition. Here is the slab of Elrathia that I showed you earlier, 3 different specimens. This is a side view of this specimen right here, edge on, and you can see very clearly how this concretion is formed. And it has a very peculiar kind of fabric to it. It is a kind of fabric that we refer to as cone and cone. And as it turns out, we have discovered that the cone and cone in these concretions is always preserved on the stratigraphically downside of the concretions. So we can use these concretions as indicators of the stratigraphic top direction, for example, or the stratigraphic down direction, if you will, of the rock units. It also suggests that the limits of the decay zone, if you will, the decay halo around the trilobite at the time that it was being decayed shortly after its death, actually extended down probably into the sediment just a little bit. All right, what else? Yes, so one remarkable thing, among several remarkable things, I suppose, about the concretions from the Wheeler and the Margin concretions is that the trilobites are commonly preserved complete, or that is to say, articulated. And as I said, they are almost always found on the upper surfaces, the stratigraphically upper surfaces of concretions. Now, the discoveries that I'll talk about in a moment or two were actually preceded by our earlier recognition, the work that I'd done in collaboration with my PhD advisor, Dick Robison at the University of Kansas. And in a discussion or a systematic work where we described a large number of new species and genera of trilobites from the Wheeler Margin interval in Utah, we discovered that the trilobites preserved in these concretions have some very distinctive patterns. And one of the most obvious patterns that we see is that some of the trilobite exoskeletons are incomplete, and that is to say they're incomplete with the free cheeks, which are these things right here, missing, or one or two of them may be missing. And that's what you see in these examples here. The other pattern we see is that the free cheeks are on. And when we find the free cheeks on, we often find that the central areas of the skeleton, the area we refer to as the axis, commonly shows crushing. Whereas when we find the skeletons missing the free cheeks, or at least one of the free cheeks, that the central area, the axis, always seems to be inflated. It became fairly obvious that what we were looking at here was an unambiguous way to distinguish between molts of trilobites, molted exoskeletons, ones that were shed in the process of growing, as opposed to corpses or the remains of creatures that had just recently died. And so when we find the free cheeks are missing or displaced, that's an indication of a molt, for example. Sometimes the cephalon or the head shield itself is actually displaced a little bit. But the most important thing about this is that the axis is not crushed. The axes in the molts are fully inflated. And this is a pattern that we found not just in the concretions from Western Utah, but this is a much more widespread pattern, not just in the Cambrian, but in fact through much of the Paleozoic. So on the other hand, corpses, the things we've interpreted as corpses, always have the free cheeks in place. We have never found specimens with the head shield or the cephalon displaced. They're always intact like this and fully articulated to the anterior part of the thorax. And in nearly all the examples that we've discovered from Western Utah, we found that the glabellas, or the portion of the head shield which overlies the stomach cavity, is crushed. And we reasoned that the reason for the crushing of the glabella, as you see in this specimen, and in this specimen, and in this specimen, the reason for the crushing of the glabella is we think that at the time of burial, the intestinal tract was fluid filled, or that is to say that the digestive tract, including the stomach, was fluid filled, whereas in the molts, it would have been empty and easily filled up with sediment. And so the sediment provided support, which at the time of compaction of sediment would have essentially buffered against crushing of the axial area. And then in the specimens that have fluid-filled guts, because there was fluid in the digestive tract of the trilobites at the time of burial, those specimens simply crushed at the time that sediment compaction took place. The implication of that is that with a fluid-filled gut, that you've got the digestive system available at the time that the specimen is undergoing early mineralization, which would lead to the fossilization ultimately. And so taking a look at some specimens like these two here, this one doesn't show too much of that's very obvious perhaps, but this one over here certainly does. This one is broken open at the crushed glabella, and what you're looking at here is clearly the stomach of the trilobite. If you follow that down a little bit, you can see that same coloration pattern, and that coloration pattern there, that sort of brownish reddish color, is due to limonite, which is basically oxidized from pyrite, which was lining the gut at the time of preservation. We can follow that pattern of color all the way down to the base of the axis right down here, and it's pretty obvious that there's crushing all the way through the gut tract. And so this gut tract is probably preserved inside this concretion. And if we look at this specimen over here, which doesn't have the gut broken open like the one on the right, we see the same basic pattern. The gut tract is broken all the way down to the end. And that pattern simply doesn't exist in these other specimens. If we look at the other specimen up here, it shows something pretty similar, at least the stomach seems to be crushed. So what we did next was to take specimens like these two here and put them under a CT scanner, getting an X-ray view of these specimens, and lo and behold, we discovered what the insides of these trilobites looked like. some of the software that some of the hardware and software that we use include things like this. And this is the same instrument that Prescott Veda talked about 2 months back, I guess it was. This is the main machine that's in the School of Earth Sciences at Ohio State. And it's a Neurologica a medical XCT scanner. And in the images that we obtained from these were resolved using ImageJ software. So those two specimens that you saw before are shown again here. This time they're reversed. And so this is the specimen that showed the open gut, the open stomach area. And this is this is just one of the specimens. This is the one that showed the open stomach cavity. And then this is an XCT scan of the same specimen. And here it is again. Actually, you're looking at something a little bit different here. The opening here extends up to about here. So this bright white structure that appears in here is actually the hypostome of the trilobite. And you can see the hypostome pretty much covers the ventral side of the stomach cavity. You can see, I think, fairly clearly that the stomach cavity extends up to this region here, and it's quite clear that you have a gut going all the way down to the anus here. Here's a little explanatory diagram showing precisely the same thing. The intestinal tract, sorry, the digestive tract begins up here in the stomach or crop and then continues down to the end here. And what we've got then is clearly a corpse. And it's a corpse that shows a crushed glabella, which sort of belies the fact that you have a corpse, and a digestive system preserved in 3 dimensions. That digestive system was probably early on in the preservation history of this creature, lined with pyrite, iron sulfide, and that has later shifted over to limonite, which is oxidized iron. Now, when we take a look at some of this material under the scanning electron microscope, we discover other interesting sorts of things related to the preservation history of the exceptionally preserved parts of this trilobite. And so what we have here is a look at the trilobite exoskeleton. I think you can see that there are a couple of fairly distinctive little segments here, thoracic segments. And then if we look at the edge of one of these segments, like right here in side view and much magnified view, now note the scale here, that's a 1 millimeter scale. We can see we can see what the cone and cone material underneath the exoskeleton looks like. And so that cone and cone probably developed as the biofilm, which was responsible for decaying the soft material of the trilobite, was being mineralized up somehow. And how was it being mineralized up? Well, if we take a look at this interesting sort of bubbly looking stuff over here, we get a better sense of what's going on. And so what you see in magnified view then is this. In highly magnified view of this trilobite, note again the scale here, this is 10 micrometers. What we see is a series of framboidal pyrite blobs that are clustered together. And what we think these represent is pyritized or fossilized microbes. And these are microbes that look like they are bacteria of some kind. And it is very likely that these are a group of microbes that could, as we say, autolithify. They had the capacity under the right geochemical circumstances to cause the precipitation, mediate the precipitation of iron sulfide precursors, leading ultimately to pyrite mineralization. So in some of these specimens, we also see strands of other microbial organisms, such as fungal hyphae, for example. And it's probably likely that it was a combination of fungi and maybe some bacteria, perhaps some other things, that were responsible for the decay of the trilobite soft parts. but it looks like some of the bacteria, for example, also were involved in causing autolithification to occur, and in mediating the process of early mineralization, leading to fossilization. How fast does that process take place? Well, we know from laboratory experiments that the soft parts of modern arthropods decay completely in marine water within about 7 to 10 days. What that means is if we are getting the guts preserved, if we are getting the digestive tracts preserved of trilobites in the Cambrian, in order for them to be preserved, the mineralization process, this autolithification process here mediated by microbes, must have taken place within about a 7 to 10 day window, meaning that these things were mineralized up and ready to just preserve long enough to become fossils. That process basically was over within less than 2 weeks. That's how fast fossilization, or at least the early stages of fossilization, seem to occur. All right, so let's take a look at another spectacular example of preserved soft parts and other non-biomineralized parts in a trilobite. This one comes from the Margin Formation, the unit that overlies the Wheeler in western Utah. This is a specimen that's preserved on rock here, and this is what the specimen looks like in side view on top of the slab that it was found on. It was apparently found in a condition something like this. And what we, in removing the specimen from the rock, this is what we see. This is the dorsal side or the back side of the trilobite. And this is, it looks a little crushed here. I think you can see very clearly that the glabella is crushed, which suggests, of course, now that we know what this means, this suggests that this was a corpse at the time that it was buried. Here in the upper right is another specimen, a complete specimen of the same species. This one does not have a crushed glabella, it appears, or barely crushed, perhaps. But it does show what the complete skeleton looks like, and this one here obviously is missing part of the thorax and in the pygidial regions. But this is what the whole thing should look like. If we flip this specimen over, this is what we see. That is to say, the way it's preserved on the rock. This is what we see. We see the outline of the trilobite here, and we see a bunch of sort of blobby looking things here, which are masses of calcite. And masses of calcite, presumably around some sort of non-biomineralized material. So let's take a look at what this thing would look like under after CT scanning. And with the CT scan, this is what we get. This is the appearance of the trilobite essentially in a three-dimensional scan, and it's remarkable. It's one of the most spectacular examples I think I've ever seen of the anatomy of a trilobite. And so what we are looking at here is the axial area of the trilobite. The front of the head is up here. This is the stomach cavity here, very clearly exhibited. And then we see kind of sporadically down the rest of the axis here in the thorax, we see the intestinal cavity, or so-called midgut. I think it's most clear right through this interval here. What we also see is an incredible array of appendages of the trilobites, beginning with the antenna, which is right here. The antenna, we can tell this is the antenna because it looks like a stack of little coins, for example, just like the antennae of a bee or a wasp or a fly or something like this. And we can see that the antenna connects to this point right here. That is the antennal articulation point on the skeleton, on the underside of the dorsal exoskeleton. It articulates here, and you can see the antenna extends from that point and curves around right up through here, and it seems to be broken. Then we can see some of the post-antennal appendages as well. I think the larger ones are more obvious. These are the big swimming legs here. but the appendages of trilobites, as most of you well know, are biramous, meaning they have two branches. One is an inner branch and one is an outer branch. And the outer branch, sorry, the inner branch is this large thing here. Then the outer branch, you can see in a couple of spots, and I think maybe up through here, you can see them perhaps better than other places. They look rather comb-like, and there's some other spots where they show up in perhaps less detail, I suppose. But this is a nice example up here, for example, of the respiratory branches of the trilobites. And then again, of course, the margin of the concretion, we think, corresponds to the edge of the microbial biofilm present at the time that the animal was decaying in sediment. Well, that's a taste of what we have found on trilobites. Let's take a look at a different style of concretion formation, and that's the siliceous concretions that we find along the Georgia-Alabama border area in southeastern United States. And these come from the Conasauga Formation. They are a little bit older than the things we've just been looking at. Most of the things we've been looking at anyway, but more or less in the same general part of the Cambrian. They come out of the Merovingian epoch. When very early on in North American paleontological history, one of the great early paleontologists, Charles Walcott, described in 1898 these fantastic siliceous concretions from Alabama, and he referred to them as star concretions. And these things range in size from a couple of centimeters up to usually not more than about 6 or 8 centimeters. Occasionally they're a little longer than that, a little wider than that, but not much more. And they have fantastic shapes to them. You can see why he called them star concretions, and he named them Bruxella alternata. Well, these star concretions captured the interest of people, I think, very early on, and early in the interpretation of these things, they've gone through a number of interpretations, but early in the interpretation of them, Walcott actually suggested that these are fossilized jellyfish. And for many years, these were used as standout examples of fossil jellyfish from the fossil record. Later, it had been suggested by Adolf Zeilacher that Bruxella actually might be a trace fossil of some sort. There are all kinds of interesting ideas about what these things were. Well, one of the things that Walcott identified when he described these things as jellyfish is he actually found that some of them had spicules associated with them. Spicules preserved from spicules that came from sponges, a special kind of sponge, the glass sponge group, the hexactinellates. Well, that suggested to us that some of these concretions, at least, might actually have a different origin. That is to say, they might not necessarily be jellyfish. And so, what do these things look like? Well, there are a number of ways of getting at the morphology of these things. One is to simply look at the external morphology, and this is actually a specimen of one of these star concretions that's slightly polished a bit. And notice that it has basically what appear to be radiating lobes, and then sort of a central area, almost like a hole or something like that, a mouth-like area in part. Another way of looking at it is to slice it open on a rock saw. And this is what one of these concretions looks like when sliced open. There's a large, what appears to be central cavity in it, and then radiating canals that extend from it. Sometimes when you slice these things open, you see spicules inside, and these little white dots in here are the sponge spicules that Walcott alluded to way back in 1898. Now, all of these are rather destructive kinds of preparation techniques, and the information that they yield is, to some extent equivocal. But XCT scanning provides a greater sense of the morphology of these creatures, and I think leads to a fairly unambiguous interpretation of their origin. And so here is an example of an XCT scan through one of these concretions, and what you see is bumps on the outside and all the sort of stuff we've been talking about already. But on the inside, look what we've got. We have very distinctive cross-shaped spicules, which are distinctive of the hexactinellid sponges or the glass sponges. And if you reconstruct one of these things in 3 dimensions, this is basically a three-dimensional series of slices arranged 3-dimensionally for a sponge. And what we're looking at here is, I think, a complete example of a sponge, showing the spongin, the material that is essentially the living tissue of the sponges. And you can see what appear to be spicules in places. I think it's all fairly clear that this thing here, compared to an example like this, suggests that, in fact, we have a sponge origin for these star concretions, these star cobbles in the Conasauga Formation. And so just in summary then, these 2 different sort of separate examples of concretions from Cambrian strata give us information about how organisms fossilized early on, how their exceptionally preserved parts came to be exceptionally preserved. We know that the exceptional preservation took place quickly in these microbial decay halos around the decaying organisms, and those decay halos ultimately became the concretions. And we know that this process took place incredibly rapidly. All of this mineralization of the softer tissues, the non-biomineralized tissues in the creatures, must have taken place within probably about a 7 to 10, maybe 14-day process. Probably not much more than that, at least for the early stages of it, but certainly well before compaction, collapse, or decay of the appendages could have taken place. We see 3-dimensional anatomy, and more often than not in these concretions when we find corpses. And in some instances, we can even find examples of fossilized microbes that were probably responsible in part for the decay and ultimately the preservation of the exceptionally preserved macrofossils.